Alright. Okay. Students, in this video I am going to explain transcription. In the earlier video I explained you regarding central dogma of molecular biology. In central dogma of molecular biology, it describes about a two-step process. One is transcription, another one is translation, where the information flows from DNA to RNA, RNA to proteins. Now, today in this video we are going to see the transcription process, that is transformation of the information from DNA to RNA. So now let us see the first step, that is the transcription process. Transcription is the first step in the gene expression. What is gene expression now? So what is gene expression? Gene expression means, for example, you just observe the central dogma diagram at the left corner. Just consider this is DNA, and somewhere in the DNA it contains some genes, and these genes are transcribed to form what is called RNA. This process is called what? Transcription. Then this RNA's information is transferred to form what is called the protein. This process, what happened? The information in the genes is passed down to form proteins. This is called gene expression. So transcription is the first step in gene expression, in which information from a gene is used to construct a final product such as protein. Okay, the goal of transcription is to make an RNA copy. That is what we just have seen. Okay, for a protein coding gene, the RNA copy, this is the RNA copy or transcript, carries the information needed to build a polypeptide. Eukaryotic transcripts need to go through some processing steps before translation into proteins. That is what the transcription process. Okay, so let me explain here in this diagram. Here in the first diagram, transcription, this is what I am going to explain. At the top you could see a double helical structure, DNA. So some part of the DNA has magnified, and you can observe there the complementary base pairs there. A is bound with T in another strand, T binds with A, G triple bond with C, A double bond with T. Likewise, so many nucleotide complementary bases are there. So this is what is called DNA. And now you observe the second diagram there. So in the second diagram, what happened? The bonds between two strands are broken down. That means the double bonds between A and T and triple bond between G and C are broken down. Then what happened? Two single strands are formed. This is one single strand and this is another single strand. So two single strands are formed, and one single strand is used to make what is called RNA. So you could see here, this is RNA that is made out of a single strand DNA. Now you just observe the base pairing in RNA. For example, in DNA, if there is T, in RNA there would be A, and if there is A here, and U would be there, and C, G. What is the difference between DNA and RNA? Only one simple thing is there. In DNA, thymine would be present, and in RNA, uracil would be present instead of thymine. So this is the RNA how it is prepared by transcription. Finally, this RNA that is prepared comes out of the DNA, that is called as transcript, and the entire process is called transcription. And later, what happened? The transcript is used for the formation of polypeptide or protein subunit. This is what we have seen. This is the process of what is called transcription. And now we are going to see another one, that is what is called RNA polymerase. The main enzyme involved in transcription is RNA polymerase. So RNA polymerase is an enzyme, that is the enzyme involved in transcription process. We will see how it works and all. This enzyme uses a single stranded DNA template to synthesize RNA. That is what we have seen there, and we will see stepwise manner in the later. So specifically, this RNA polymerase builds RNA strand in 5' to 3' direction. Very, very important thing. So we know that DNA has two strands. One runs from 5' to 3' direction. The another one runs in 3' to 5' direction. Both are anti-parallel to each other. That is what we had. But here the RNA polymerase synthesizes the RNA strand in 5' to 3' direction, and it adds each nucleotide at the end of the 3'. Okay, this is all that about what is called RNA polymerase. That is the only enzyme that is involved in the transcription process. Now we are going to see the stages of transcription. Okay, this explains how actually transcription takes place. This transcription takes place in 3 stages. The one is, initiation. The another one is elongation. The third one is what is that one? Termination. So we see this how transcription takes place by taking an example of a bacteria. Now the first one what we are going to see is called C is that one. C is initiation. That is what we are going to see. And now RNA polymerase. This RNA polymerase binds to a sequence of DNA called, what is that one? Promoter. It binds a sequence called promoter. Promoter is present at the beginning of the gene. Each gene has its own promoter. Once bound, this RNA polymerase once bound, what it does? It separates the double helical DNA into 2 separate single strands. Okay, we will explain that one in a diagrammatic fashion. Here in the first, you just observe the top diagram where double helical DNA is drawn. One runs at 5' to 3' prime, the another runs at 3' to 5' prime. So here this region, this entire region is a gene, and within the gene you could see a transcribed region. This is the only region where RNA is formed, and before that you could see another region called promoter region. Promoter region, every gene used to have a promoter. Why this promoter? This promoter used to have some specific site called recognition, specific sites called some recognition sites would be there, and these sites are recognized by RNA polymerase. And once the RNA polymerase recognizes the recognition sites, and simply it comes and binds on the promoter sites. Once it binds, what it does? It does, see, This is what a bubble is formed here. Have you seen this one? A bubble is formed. Two single strands are formed. Whatever the whatever the bonds in between these two strands are broken down. Now two single stranded templates are formed. Now let us see what happened. With that we come to an end of the first phase, that is initiation. In the initiation, what we have seen? We have seen the DNA where genes are present, and within the gene we have seen one transcriber region and one promoter region, and within the promoter region we observed that RNA polymerase came and bound there, and once it bound, it starts it opens the double-stranded DNA into two single-stranded templates. That is what we have seen. And now, the second one is going to be elongation. Elongation means what? The continuity. Once the RNA polymerase bounds, it starts what is called elongation. It starts elongation. On one strand of DNA, we already know that the RNA polymerase uses only one strand of DNA. The one strand is called, that is utilized by RNA polymerase, is called template strand. This template strand utilized by whom? RNA polymerase. And this RNA polymerase reads this template strand one base at a time. I will explain how it reads and all that. The polymerase builds RNA molecule out of complementary nucleotides, making a chain that grows 5' to 3'. The RNA transcript carries the same information as a template strand or coding strand of DNA. That is a non-template coding strand of DNA. We will discuss that one in the diagram. It contains the base that is uracil instead of what is that one? T, that is thymine. Okay. And now here let me explain you the elongation process, whatever we studied over there. And you already observed this one. This region is which is a promoter side, where recognition sequences are there. Our RNA polymerase bound here, and then this RNA polymerase starts moving this direction. Okay, while it is moving, what it does? It reads the template strand one base at a time. That means, what is the meaning of the word one base at a time? That means, for example, you see this is template strand and this is RNA polymerase is moving. For example, if the RNA polymerase first come across T and it incorporates, what is that one? A. If there is A, it incorporates U. If it is C, it incorporates G. Likewise, so RNA polymerase incorporates complementary bases of the template strand, but it constructs RNA, it do not incorporate what is that one? Thymine, but it incorporates uracil. So now, in that manner, what is happening? By incorporating one after another, what is happening? A single-stranded RNA is formed. The single-stranded RNA represents what? Which is complementary to the template strand, and it is same as what? It would be same as coding strand, which is non-template strand. So don't get confused. There are two strands in DNA. One strand is recognized by RNA polymerase, which is called template strand. The another strand that is not utilized by RNA polymerase, which is called coding strand. Now, with that we come to an end of the second stage, that is elongation. Finally, we come to an end of the last one, that is called termination. In termination means what? Coming halt of RNA polymerase on the DNA strand. Sequences called terminators. So DNA used to have some sequences. These sequences are called terminators. These are called terminating signals. These terminator signals come across by the RNA polymerase, then RNA transcripts won't, the RNA polymerase won't go further. It comes to a dead end. It comes to a halt. Now with that, RNA transcription is complete. Once they are transcribed, they cause the transcript. Now they cause the transcript to be released from. They cause the transcript to be released from the RNA polymerase. So once the transcription is come to halt, then whatever the RNA that has formed so far is released from the RNA polymerase. So this is RNA polymerase that is released from RNA polymerase. So you could see here, this is called hairpin structure. Once the polymerase come across some kind of terminator signals, it forms a hairpin structures so that this termination come to halt. So this is called, what is that one? Termination of, what is that one? Transcription. So far what we have seen that central dogma of molecular biology, what it explains in the central dogma of molecular biology. So in central dogma of molecular biology, we explained how the information is passed from DNA to RNA, RNA to protein. That is what we explained. And later we explained the transcription process. So transcription is the first step of gene expression. That is very important. What is gene expression? Gene expression is a particular part of the DNA is expressed into RNA, then RNA is converted into protein. This is called gene expression. Gene to RNA, RNA to protein. This entire process is said to be gene expression. That is what we have seen. And coming to the another one, we have seen the entire process. And finally we have seen, what is that one? RNA polymerase. And this is the RNA polymerase here, the shaded one, which is bound on the DNA template and start making, what is that one? RNA here. So now another one, stages of transcription. What is the first stage? That is initiation. What is the second stage? That is elongation. What is the last stage? That is termination. With that we come to an end of what is that one? Trans